Welcome, everyone. Thanks very much, everyone, for coming across to this actually pretty pleasant venue that I didn't know existed. I'm using a microphone, even though everyone in the room can hear me, because we do have quite a few people on Zoom as well. So Richard will also use a microphone, even though his booming voice is not requiring of it for the room. And we'll do the same thing, even though it'll look a bit weird. We'll use a microphone when it comes to the Q&A session at the end, which can be a discussion session. Richard is very relaxed and very keen to be able to talk to people in the room. I want to start with an acknowledgement. And we acknowledge and pay our respect to the traditional custodians of the Kulin Nation and all across the world. We recognise the traditional custodians as having a unique ability to care for country and the deep spiritual connection to it. We honour elders past and present whose knowledge and wisdom has ensured the continuation of cultural and traditional practices, and we're committed to genuinely partner and meaningfully engage with Australia's traditional owners, Aboriginal and Torres Strait Islander communities. I won't go on and on, I promise I won't eat up too much time, but I also did want to give a very short introduction of Richard here. We hugely value his time. Richard, you may or may not know, is here in Australia as part of a tour for the premiere of the movie Salt in My Soul. We went along yesterday, so Richard has been in every capital city so far and still has, I think, Sydney to go tomorrow. So this is an amazing movie, and I would thoroughly recommend to anyone who's in any way interested to have a look that you really should. It tracks the life and untimely death of a really amazing young woman, an American woman, Mallory Smith, and a hugely powerful movie. I, again, am getting kind of emotional now just from remembering what we witnessed last night. Far from being a tearjerker, the documentary about Mallory's life is in fact a hugely empowering movie, and I think it was extremely well made. And the Q&A session that followed on from that was really, I thought, very exciting. And Richard has done these sessions every capital city that the movie has premiered. Really amazing in the way in which people discussed Mallory's life. Her mother, Diane Shader Smith, was there to host the Q&A and to present the story that Mallory had documented in this video, and it was fantastic. So Richard's here in Australia for that reason, but he is here at Monash to present on CARB-X and also to discuss with us because he is a great friend of the Centre to Impact AMR here at Monash. And looking around the room and seeing that there are a lot of PhD students and postdocs who are just starting out on their scientific careers, I thought that I should make sure that I introduce Richard as someone who was you and therefore is still a future version of you. Richard did his PhD in molecular microbiology at the University of Adelaide, not so long ago. So he was a PhD student who was not sure what he wanted to do with his career. He did a couple of postdocs, I think one in Australia, one overseas in Canada, if I'm not wrong, and then decided to move into industry, had perhaps a lot of years of experience in big pharma and also in small companies, and then was recruited on as the Chief Scientific Officer for the CARB-X consortium. So I think, you know, for those of you who want someone to look at as a role model and for a bit of inspiration about where you can take your careers, he's here today. Richard will also, we've not told him yet, but we're hoping to invite him to come back later in the year to visit at Monash, and so there would be perhaps an opportunity, anyone who'd be interested to talk with me about having some sort of session, perhaps about professional development and ECRs and making decisions about your career. I'm sure we can convince Richard to be involved with that. Now I am burning up time, so I'm just going to hand over to you.
It is, ooh, that's loud. Thank you, Trevor. Thank you. And just before I start, the movie, it's funny, you know, I watched it. I was told to watch the movie before I came to do the tour because you didn't want to be emotional up on the stage, you know, in the panel. So I did, and it was sad. It's a sad movie, but I will say that each of the 4 times I've seen it since, it's getting harder and harder to watch. And the reason is because now I know the parents. I'd met them online. I'd met the mother online before, but never met them personally. And mum and dad are both here touring Australia, and to now know them as people and then understand the suffering that they went through, it makes the movie harder to watch. And so I can't believe the courage they have to share their story, but that's the power of a personal story. And they say, one person dying is a tragedy, 1 million is a statistic, but every person that dies of AMR has a grieving family, friends, and community around them. And that's what we're all here to try and fix. So I'll move on to my talk. So CARB-X, I'll give you a little bit of introduction about what CARB-X is if you don't know. And then we've been in existence since 2016. We're a global nonprofit, and we were formed out of an executive order signed by President Obama. So CARB stands for Combating Antibiotic-Resistant Bacteria. He put an executive order in place which authorized the US government to fund an accelerator or an incubator to give non-dilutive money to companies and groups that are developing in the antibacterial resistance space. New therapeutics, preventatives, and diagnostics was our remit. That's expanded since. Oops, they said right. This is all right. Now I don't know why this is not working, Terry. There we go. So it's expanded since. We have 5 funders now currently. So ASPR and BARDA is the US government. We also get money from the United Kingdom or the UK government through their GAMREF, Global Antimicrobial Resistance Innovation Fund. The German government gives us money, and then we have 2 philanthropic donors, the Bill and Melinda Gates Foundation and the Wellcome Trust is our second biggest donor. So they're the 5 groups that have funded us since 2016. BARDA and Wellcome Trust have just re-upped because they have different funding cycles. The US government is committed out to 2032, so they gave us an extra 10 years. And the Wellcome Trust works in the smaller cycles. They've given us $70 million over 3 years. So all told now, CARB-X has been lucky enough to receive a promise of $822 million US to give out non-dilutively. And people say, well, what's non-dilutively mean? That is basically a grant. So it's a cost share grant. We give money, but the company or the group has to put in a certain percentage. During COVID, the funders changed the ratio, and we were paying 90% to try and encourage VC firms to come in and invest in the space, because a lot of VC firms aren't. So that's who we are. I think I don't have to tell people here the importance of antibacterials. Antibiotics are probably the most ignored part of medicine. They've been around for years now, and yet they've been taken for granted. And we as a global society have really, I think, abused the precious gift that is an antibiotic. Every time you go in for any type of surgery, you take the fact for granted that you'll get an antibiotic that will kill any bacteria that may cause an infection. And so as the resistance problem is getting worse, a lot of these foundational surgeries and what we take, you can go in and get a hip replacement now, an organ transplant, all of those type of surgeries are being put at risk, not because of the surgeon, but because of the infection. And I tell a story of a friend in the Boston area that was getting a triple bypass. And I went in, and in his office, he had a list of all these doctors. And I joked, I said, oh, you're working out who's going to do your surgery. And his response was actually quite harsh to listen to. He said, no, he said, there's over 250 doctors in this city that I would trust to do the surgery. I'm going through the infection rates at the hospitals where I have to have my recovery, because that's what's going to make the difference. And I'm like, whoa, that's what it's like, right? You don't think about the surgery anymore. You think about the post-surgical complications, which is infection. So dialysis, sepsis, we've all seen the numbers. I think this is a paper that we talked about last night, and I know our groups here in Melbourne actually contributed some of the data to this global study that was looked at the number of deaths that were attributable or associated. Now, they're hard words to understand. So the paper came and said that there was 1.27 million global deaths that were directly attributable not to bacteria infections, but to resistant bacterial infections. There was 4.95 million deaths associated, which is sometimes hard to distinguish. I think significant impact in the low and middle income countries. I've got a slide on that in a minute. I think what a lot of people found staggering in these numbers was that the drug-resistant species like E. coli and Kleb and Staph were actually higher than TB, because everyone, and when I was going through all those years ago, TB was, I was told that if every microbiologist worked on the disease proportional to the people it killed, we'd all be working on TB. But now you can see there's 6 pathogens that are above TB. So this is a number that, you know, means something, but what does it really mean? And at 1.27 million deaths, that's a plane crashing every 2.5 hours and killing everyone on board for a year. At the 4.95 million deaths, as I said last night, for the people that may have been there, that's every 39 minutes. And so if you think about a plane falling out of the air every 39 minutes, governments would do something about that. And yet they don't do a lot about AMR because it's behind the scenes. People think they die of cancer or they die of cystic fibrosis like Mallory did, but it's not that. It's the infections behind that that's killing people. And so it's, you know, that's, I think, the issue that we need to address. So the mortality numbers are increasing. The estimates that I've heard is up to, you know, within 10 years, we'll be at 10 million. That's a death every 3 seconds. So while I'm standing here flapping my gums, you can imagine how many people will have died. So when you put AMR in context with all other deaths, not just like HIV and tuberculosis, you know, I've got, you know, HIV and AIDS on this slide, but if you look at, you know, heart disease, stroke, dementia, you can see where AMR comes. If you look at just the dark red, where it's attributed to the resistance, but if you look where it's associated with, you're actually the 3rd largest cause of death in the world. And yet we don't even talk about it, right? Because it's often a complication. So I think every conversation you have as microbiologists or as researchers around AMR is a good conversation. I think the general population now, because of COVID, a little bit more understanding of what an infectious disease is. A lot of them probably don't know the difference between a bacteria and a virus. So that's your job to help engage and educate them. But I often say, don't let a good pandemic go to waste. And I know Melbourne suffered during the pandemic with lockdowns, but this is an opportunity to turn that into a positive message and say, this is another pandemic that is coming that we need to address. So obviously, Australia is a high-income country and has a very good healthcare system. If you look at the death toll by geography around the world that this study showed, you can see that sub-Saharan Africa and South Asia are where a lot of the deaths are. And the dark blue bars that take up most of this graph are the neonates. Now, neonates are the first 30 days of life. And so if you actually look at that, this is the rates of neonatal death around the world. So per 100,000 neonates, over 25,000 of them are dying. So that's more than 1 in 4 neonates die within their first month from AMR. And that is a horrifying, staggering number. And most of that's in, again, low and middle income countries. They take most of the burden. But if you look at ages 5 and above, so that's, you know, 5 to 100, 69-fold less for the next post 1 year or post 1 month of your life. That's the difference. So getting through that first month, especially in those countries, is hard. And so CARB-X has calls on at the moment. After seeing this, the funders have put out a call for neonatal sepsis vaccines because it's clearly a huge unmet medical need. So what does CARB-X do in its day-to-day other than sending me to Australia to talk? We are a global partnership fund. We fund all over the world. And so the funders that give us money have no geographical restrictions. We fund the best science we come across, no matter where it comes from and no matter who it comes from, be it academic, a small company, a large company, or a consortium. As I said, we've had 822 million committed out to 2032, and we work on preventatives, not just vaccines, other preventatives as well, therapeutics, both traditional and non-traditional, and also diagnostic products. And that's platforms looking at both identification and AST, or antimicrobial susceptibility. We are the most scientifically diverse development portfolio. and we treat it as a portfolio. We've had 1,163 applications before the current rounds, and we've only funded 92 projects. So that means two things. One, it's hard to get into CARB-X. That's a funding rate of about 7.8% or something. So it is hard to get funded. We think we offer benefits that I'll talk about when we do fund groups. But it also says that there's a lot of activity out there that needs funding. And I've got a slide on that. So this is where we sit in the chain, if you like. And it really is research and development, especially for the academics in the room. Research and development is a very different world. I didn't realize that until I went and worked for a company. Very different mindset and experiences. You need the basic research. That's the most important thing you can have. That's the foundation. And there's a lot of people that fund basic research, including MRFF and NHMRC and ARC here in Australia. But it's that translational piece, taking that basic research through where CARB-X plays. We fund hit to lead through to first time in human, or the equivalent stages in diagnostics. And we used to have some colleagues in that space, and they are rapidly falling out. So Novo Repair used to make dilutive investments in that space. They've now paused their investment. And the IMI AMR accelerator is focused basically on TB, and new drugs for bad bugs has stopped. So CARB-X is really the only push mechanism that is left at any scale at that stage. And our job, we see it, is to make products suitable for late stage development money, because there's not much money in development of late stages either, with all the big pharma getting out. You have to look at other groups that will help fund phase 2, phase 3 and registrational trials. And so BARDA is an advanced developer. HERA is being stood up in Europe. GARD-P, I think we all know about them, out of Geneva. And then the AMR Action Fund, which is the big pharma, have now put in 1.6 billion, I think, total, to help develop and commercialize products. And they've been around, I think, 3 years now. It's the first year of the pandemic, and they've made, I think, 3 investments. And even their CEO has come out and said he did not know that the pipeline was so thin that they're having trouble finding products to truly invest in to come up with differentiated medicines. So that's what he says here. That's Henry Skinner. He was chairman of the board of the small company I was at, involved in the development of linezolid. So he runs the AMR Action Fund. And so I think, and I said this last night, so at least some people heard it, the funding is one of the biggest, and I don't want to stand up and say, oh, you guys all know how hard funding is, but there's some aspects of funding that I think are really important. It's not just the amount and the availability of it, it's the longevity of it. Giving people 1 to 2 year grants does not help, because then you've got to find people again, especially in an academic institution, and no one likes living grant to grant and all you do is write grants. And so it's longevity of that funding that's important. I also do some work with the WHO, and the clinical pipeline and the preclinical pipeline is very, very thin. I think we all know that. And I often think back to my days at AstraZeneca when I was waiting to go into the senior management team to give a presentation. I was going through my templated slides and looking at my portfolio slide of where we sit with the competition. And I was sitting next to a cancer colleague and he was doing the same. And he looked at my slide and he said, oh, is that your target? And I said, no, that's the whole antibacterial pipeline from phase 1 to phase 3 in the world. And he showed me his that had like 60 projects on it. And he said, this is my kinase target. And that's the difference, that his slide had 60 programs in clinical development against a single enzyme. And I had seven projects against all bacterial that were in development. And that's the difference between the funding schemes. So you'll see the WHO is gives the same perception on who is doing the work, and I've got a slide about that later. So there's been a lot of white papers written, and they're good, but just writing more and more reports doesn't always get you where you want to go. But basically, several large groups have written these white papers to say, okay, what do we really need in this space? And the average is about 6 innovative new treatments with differential performance characteristics that can make an impact to patients per decade to keep ahead. I'd like to think that, 10 is a better number, but 6 is, would be good considering how many we get now. And so this is how many you need to get 6 because of the probability of transition and success through the different stages. To get the 6 out the bottom of the funnel, you need to put about 6,000 in because that's how many fail. And so if you're working on a project and it doesn't seem to go very well, you have a lot of good company because a lot of people fail. And so if you want to be right 98% of the time, don't work on anything, basically. But if you want to make a difference, keep working on that because you might find the one that makes the difference. So we fund in the early stage product development, and this is now data that's been taken from the Global AMR Hub run out of Berlin. And we look to see, well, how much money is needed to progress those projects to how much is actually committed and available. And so basic research, they say in the next decade about, 3 billion. Gets a bit more expensive in early stage development where you do all the tox work. So that's about 5.6 billion. And then advanced development and post-approval, because so many fall out in the tox stage, it's about 3.5 billion. So if you look at that's what's needed, and then you look at what's available, you can see the basic research, based on the AMR Hub data and talking to other groups that fund, it's about 2.9 billion. So you're close. 2.6 in the bottom of the hourglass now, compared to 3.3 is within the ballpark. But if you look in that middle, the largest funding gap is in that translational research. 1.8 billion committed over the next 10 years, where we need 5.6. And so that's a big, big gap. And that's where most products will fail, sadly. So currently we have 39 projects in our portfolio as of the making of this slide, which was back in October. We have 21 therapeutics. They're all new, they're all innovative, and over about 2/3 of them, exactly 2/3 if my math's right, are non-traditional. And so we do have a focus on high risk, high reward products. We don't tend not to fund me too, just incremental improvements. While they can make an impact on patients, resistance often erodes them before they even get out on the market. We have 11 preventatives, 7 of which are vaccines, but we also have phage and microbiome, live bacterial therapeutic products in that space. And then 7 diagnostics in our portfolio. One of them has graduated, I know, since this slide was made. So when that Graham paper came out and said, okay, this is how many people are dying and this is the indications they die from, obviously our funders want to know, well, what are you funding? How does that match up? Do you meet that need? And so we've mapped all the 92 projects that we have funded across the indications that we supported them for. And sometimes it's better to be lucky than good, but most of them come up against respiratory indications and BSI, because we did fund a lot of focus on gram-negative pathogens, and that's where you see a lot of gram-negative infections is in the respiratory tract. I will point out on this slide just at the bottom, gonorrhea, that was not included in the big report in 2019 that came out last year, because gonorrhea doesn't cause a lot of deaths. There is some death through disseminated gonorrhea, but gonorrhea has more of a morbidity problem rather than a mortality problem. And so it wasn't included in that, but you can see that we actually have funded gonorrhea problem projects, both in all 3 pillars, because it's a huge public health need, and it is in Australia. I think anyone that follows AMR in Australia would know that drug-resistant gonorrhea is a big problem here. Co-infections with chlamydia, mycoplasma is making it really hard to diagnose and treat those patients effectively. And so from a surveillance and a treatment perspective, gonorrhea is a big problem. We have partnered, we're an accelerator ourselves, and people say, well, why do you have, acceleration partners? Because part of the job that we do is to try and understand the landscape of science that's going on around the world. And so we have acceleration partners around the world that help us with that. They provide expertise as well as local knowledge about the ecosystem. And so we are working, CARB-X is working with MTP Connect here in Australia to try and build out what is the AAMR net, to make that a fully fledged accelerator within Australia and the South Pacific. And hopefully that would be a benefit to both the Australian ecosystem, but also to CARB-X. So there's been 38 applications from CARB-X from Australian groups. We've funded 3 awards, 2 from the University of Queensland and then a diagnostics company in Sydney. But the pitch to anyone that will listen here in Australia is some of the benefits that an accelerator would bring, and they're listed there. And I think we've also put money through some of our other ex-Australian companies that do their clinical trials here in Melbourne and in Adelaide. The clinical trial network with its rebates in Australia is very attractive to small companies because they get money back. And so several of our companies have invested in Australian science by doing their clinical trials here, which is another advantage of having an accelerator because they can tell them of the benefits. So our goal is to balance novelty with risk. I've talked about novelty. I think if you're going to make a step change, you need to come up with non-traditional ways and think about new paradigms on how to treat these infections. I think some of the groups here in the Centre to Impact AMR here at Monash are actually thinking about some of these novel technologies. And I think bacteriophage is one of them. And, you know, it's been fascinating to watch the resurgence of bacteriophage. Many years ago, when I was doing my PhD, bacteriophage were really just N13 cloning so that you could sequence. Back in the days when you had to make phage preps to sequence and mouth pet P33 into the acrylamide gels. That's how old I am. So phage have come a long way, even in you know, my career, and it's wonderful to see that. I think what will really benefit, while we're seeing compassionate use cases truly benefit some patients now, I think some controlled clinical trials to understand the patient populations, the species, the antibiotics you give in conjunction, and the duration and regimen of therapy. Some of those controlled clinical variables will be really informative to help turn phage therapy into a more empiric use therapy and not just compassionate use. And hopefully that's where the field is going. We do emphasize, and I gave you the statistics as to why, we do emphasize performance characteristics in our products that will benefit LMICs. Some of our funders will only fund activities that will directly benefit LMICs or low and middle income countries. And this is obviously things like cost of goods, stability in high humidity and high temperature, dosing regimen flexibility. You can't always get rural communities to come in multiple times. And obviously robustness to existing pools of resistance because of the burden of disease in those countries. That's where a lot of the resistance originates. The other big thing that CARB-X has maintained from the start is this stewardship and access. So stewardship and access is, I hope people understand stewardship and access. Stewardship is using the right drug at the right time for the right patient. Think of it as if you've got an upper respiratory tract infection and you think it may be a bacterial infection, could be a virus, but you take an antibiotic anyway, that's bad stewardship. If it's not a bacteria and it's a virus, you don't need an antibiotic. Access is just, does the right patient get access to an antibiotic? And access issues exist in obviously poor countries, but they also exist in countries like Australia. And I used the statistic last night that of the last 19, I think, antibiotics that have been approved in the US, you can buy 3 of them in Australia because companies don't promote them here because there's not enough money to sell them. And so to go through all the regulatory approval and then the commercial arm, it's just not worth it. So even access to some of the newer drugs, you can't get in Australia. Innovation, stewardship, access, you can have, it's 3 legs of a stool. You need all 3 before the thing is useful. And so before the pipeline is useful, you need all 3. And that's where pull mechanisms, and that's a whole different conversation, but basically a pull mechanism is a way that governments and society need to de-link the amount of drug you sell, which causes resistance, to having it available when you need it. So again, think of an insurance policy. You hope you never use it. You hope your house never burns down, or you hope no one ever burgles your house, but you pay your insurance every year in case. That's what antibiotics are like. You've got to have them there in case, but you hope you never need them. And so CARB-X actually wrote stewardship and access policies into our contract. So every group that we fund has to sign on the contract a contractual obligation to actually build a stewardship and access policy should their product that we helped fund in the early stages ever make it through to registration. And we'll be long gone, CARB-X will be long gone from their life by the time it gets registered, but the contractual obligation has bleed through, and Wellcome Trust has step-in rights. So if you don't do it, Wellcome Trust can come and say, okay, we want to take your product into LMICs now. So this plan is freely available on our website. We hope companies that we don't fund that are committed to understanding the impact of stewardship and access take advantage of it. And so it's freely available for them to model if they feel like it, but all the CARB-X companies are contractually obliged to it. So people ask me, well, what's the landscape been like in the last 6 years since CARB-X started? So I've put a couple of slides together here. As I said, 7.9% of the almost 1,200 applications have been funded. We have received applications from 39 different countries. It was, in the beginning, it was very US biased. I think because BARDA made such a big deal and because that's where a lot of research happens, that has decreased. So in our last funding rounds, it was below 50%, which is actually good because that means you're sampling the rest of the world. There are certain regions of the world where you can see we don't get applications. We'd hoped that the current funding rounds would have changed those demographics a little bit. They don't seem to have moved the needle much. And so we're actually actively reaching out now to research groups and some of the scientific accelerators in Africa countries and in South America to try and find out why it is. Is it because they don't know about CARB-X? Is it because they don't do the work? Now, obviously, high-income countries need to carry the bulk of the load. It is a global problem. If you don't fix it in the LMICs, you're going to have the problem in your country. But it's also, I think, important to understand because anyone can have a good idea and anyone can, have a project that's going to benefit patients. And so we need to make sure that our message is getting out and that they are supported enough to put in an application that may be, the next drug. So we are doing that additional outreach. So that's something we've learned. Western Pacific, obviously that's of 80 of those, I think almost half came from Australia. Most of the people that work in this space work in really, really small groups and small companies. And that is, I think, the most dangerous thing into our ecosystem is how fragile it is. So almost half of the groups that we fund are less than 10 people. Now that has two impacts. One, they're usually single asset, which means if that asset doesn't work, the company folds. And I've done this analysis both on our applications, but also for the World Health Organization. In a rolling 3-year period, about 50% of the companies no longer exist. And so that's the turnover you get. Now, that's hard for your superannuation, but it's more devastating for, the field. I think that there's no resilience, and that comes back to the longer term funding that's needed. So you can actually have more than one asset in your portfolio as a company and actually train those people. I was lucky enough to train in a large pharma. Small companies don't train people. They hire to execute, they don't hire to train. And so I think the loss of human capital is huge in this field. And to put that in perspective, when AstraZeneca got rid of 120 infection researchers in May of 2015, that was about a 3-week gap before Merck bought Cubist up the road and fired all of them. So there was about 230 infection scientists on the street at the same time in Boston looking for jobs, which is not a good environment. I don't know all the Cubist people, obviously, but of the 120 from AstraZeneca, I know of 5 that have remained in infection. All the rest have gone to other therapeutic areas. And that's the human capital that you're losing. No one's training the next generation. And so, I welcome the opportunity to come back and talk to the younger people in the room about, directions your career can take, because it's really important that the next generation learn what to do and how to do it. And that's something that hopefully I can pass on, because as my daughter tells me, oh, Dad, you're at the age of your career, you should do something that's good for the next generation, because you're not giving us anything else. So, so I see that as part of giving back now, to help support the next thing. But the attrition is horrible, and small groups carry all of the risk. So this shows in a graphical format what I told you before about the risk of projects failing as they get closer to man. So this is just the applications. You can see all the applications that have come in. There's quite a lot in Hit to Lead, which is the first phase that we fund. That's, do you have in small molecule sense, do you have an inhibitor that inhibits the bacteria? As long as you've got wild type activity, you don't have to have shown it in an animal model yet. That's Hit to Lead. And that's where most of the programs are. But by the time you get to phase 1, which is more expensive, the number of applications drop off significantly. And that's because typically they all die in preclinical, which is when you do your tox. And because you have to give so much of these drugs, that's where you lose so much. And I used to think that, once you get into drug development, a lot of those therapeutic area agnostic disciplines like toxicology and all are the same, whether you're in infection or cancer or CNS. I can tell you it's not, because if you go to a formulation and a toxicologist and you tell them that you want to dose up to 2 grams per kilogram in a mouse or a rat or a dog, they go, what? How are we going to make that soluble? Or how are you going to make that much at an early stage of a program? And so a lot of those therapeutic area agnostic disciplines in a pharmaceutical company are still very infection specific, because the challenges of formulation and dosing for infection products are just so unique. This is one that I think I did not expect this. And I first saw this when I started doing the pipeline analysis for the World Health Organization. And then I went back and did it and looked at the CARB-X applications. And you see the same thing. There is this real shift towards narrow spectrum products. And that is good from a stewardship perspective. It's good from protecting the microbiome perspective. It's bad from a commercial perspective, because drug companies like, just use my drug and it will cure everything. But it's also, I think, a side effect of both the CDC and the WHO publishing their priority pathogens list, because they put out a list of bacteria with the right intention, saying these are the most important pathogens, these are the ones you need to do research on. And a lot of people in new and upcoming companies or groups would look at that list and say, oh, carbapenem resistant antibacterial and Enterobacteriaceae, if I get a product that's selective for Citrobacter frondii, I've got a drug. Well, no, you don't, because there's no way to develop that. There is no way to develop a Citrobacter frondii carbapenem resistant drug. You just can't develop it. And so I have implored with the WHO that their list needs to come with an instruction guide on how to interpret that list and how to use it. And hopefully when the PPL, the priority pathogens list, gets renewed in mid-year this year, they will come with a little bit more context on how to interpret that and what pathogens you need to cover per indication that can actually make it possible to do a trial in a reasonable amount of time and a reasonable amount of money and get it approved. And so, but I love personalized medicine. I think that's the way of the future. I think that'll obviously put a stricter need on diagnostics. And I think the technologies are there. We need the next generation of doctors to embrace molecular diagnostics and not, be older doctors. We've been doing culture for 40 years, and I've always treated on my gut. Well, we've got the tools now to understand diagnostics, but you need to put that into clinical decision making. So I think that will come, but we really need to understand the regulatory pathway for species specific, and also understand how to treat specific indications and the polymicrobial pathogens that may be in there. Our funders always ask us, well, where does our money go? And as a non-profit, you hear stories about how much money goes towards overhead and administration. So I was happy to see this pie chart. So 80% of our money goes directly. We don't even touch it. It goes directly to the people we fund. And another 15% comes from like in-kind NIAID support. SMEs are not that what we call them, annoyingly. We call them SMEs, are not small and medium enterprises like most other people. We call them subject matter experts. We build a team around every group that we fund. We build a team. We call it a CARB-X support team or a CST. And that's because a lot of small groups or small companies don't have all of the disciplines they need across the entire development pathway. If they're at hit to lead, they don't have a clinical or regulatory person because they don't want to pay them that yet. And they've only got one asset. And so from a clinical perspective, they need to be asking those questions though, and having those conversations so they know what preclinical work they have to do to make that package as robust as possible. So when they get there, they've got the right data package and it's attractive to advanced developers. And so what we do is we use our network of consultants that doesn't come out of the company's budget. We pay that separately to actually give them free advice and have those discussions and fill in for them with CMOs and regulatory people as they're developing their products so that they can have those discussions in a meaningful setting and start thinking about those. And so only 5% of our money goes towards BU and the non-scientific management of CARB-X. So that's a pretty good batting average. And I'm sure our funders want to see that maintained. So. The other thing we noticed a couple of years ago, and this is the company support team. I've just gone through that. Sorry, I didn't know I had that slide in. Whoops. So yeah, I've done that. The other thing we've noticed is when you give funding out to multiple different groups, a lot of them struggle with the same problems. And because you can't tell company one, well, company two's got the same problem, you should talk to them because you're bound by confidentiality. It became somewhat frustrating to give money to multiple groups to try and answer the same problem. And that wasn't good use. We weren't being good stewards of the funds that we've been given. So we came up with a concept of project acceleration tools or portfolio acceleration tools to try and answer the questions on a larger scale that we could then, as CARB-X, we could answer the questions and then give that information not just back to all of our companies that may be experiencing that, but also to the greater ecosystem if we share it at conferences and publish it. And so we started doing some of these portfolio acceleration tools. They grew organically. Actually, out of the first one was with the University of Queensland on nephrotoxicity risks around peptides. And then these are some of the ones that are either ongoing, finished, or in discussion. So I'll go through a couple of them in a little bit more detail. One is the early evaluation of pre-existing resistance. A lot of small companies don't have access to isolates from around the world. And so they don't know, they test their compound against a 50-year-old ATCC strain. They don't know whether and they kill it and they think, we've got Klebsiella activity. And then they go and see the hypervirulent Klebsiellas that are coming out of Southeast Asia and they're, greater than 128 or greater than 256. And they say, oh, that's odd. Well, I thought we could kill Klebsiella. Some companies don't want to know that because their funders might get nervous and pull their funding. And so, there's a mix there, but I think from, in all honesty, you need to know how good your compound is as early as possible. And you need to know there's pools of pre-existing resistance. And so we developed a program to address that, and I'll go through that. That bled organically into this early evaluation of antigenic variability. We have a lot of vaccine companies that don't have the bioinformatics expertise to actually look at the level of conservation of their antigens across multiple isolates from multiple different countries to see whether the vaccine antigens they're using are actually going to be useful. There's no point making a vaccine with a protein in it that you found in Baltimore, Maryland, when it's completely different to the ones in sub-Saharan Africa, because that vaccine is going to be useless. And so we now offer customized bioinformatics at CARB-X. That's my evening job. And so we do that for them as well. And we're also addressing models of efficacy. I think some of the efficacy models are good. Obviously, the thigh model for infection is really good, but some of the other ones don't have that back translation. Compounds that should work don't, and compounds that shouldn't work do. And so I think we are working with other accelerators around the world to actually build a lung model at the moment that can be, the strains can be widely shared amongst the community with a protocol and back translation evidence, so that you know what to expect in your animal model that will translate to a clinical success. And we have a couple of other models that we're interested in, including the gonorrhea and UTI ones. So I'll just go through the pre-existing resistance risk because it's quite an interesting story. We weren't sure whether groups would be willing to participate in this, to be honest. And my boss said, well, go out and ask them. So I asked all of our companies in the therapeutic space, would they be willing to give me a couple of their compounds to screen against a large panel of contemporary isolates? And all but 2 did. And the 2 that didn't had very good reasons. One was too early. They didn't have any antimicrobial activity, even against the ATCC strain, so it seemed pointless. And the other one was in phase 1. And as you know, if you generate data in phase 1, then it's got to be declared to the FDA. And so they sort of held off. So I understood both of those, but every other company said they'd be happy to. And so we worked with IMHA, which is a global purveyor, if you like. They do surveillance. They and JMI are the 2 biggest ones in the world. They sent me their list of isolates, 96,000 isolates they had collected through 2019 with no information in it. All the information that I got was, this is the country it came from, this is the age and sex of the patient, this is the hospital it came from, and this is the source, whether it came from a blood isolate or a urine isolate. I had no susceptibility data. So that's important to remember. Because of the focus we were looking at, we changed the bias a little bit. We made about 60% of the isolates from LMICs. We couldn't do it all from LMICs because you still wanted to understand the resistance prevalence in perspective of what you were seeing in LMICs compared to high-income countries. So we made it about 60% from 67 different countries, which was a very wide selection. And we also extended the program that if we saw isolates that had decreased susceptibility, we would work with IMHA to make those isolates available to the company so they could do follow-up research. And that actually turned into quite a big program. So this is the data. And so what I've done here is I've actually mapped across the x-axis is the species. So there's 5 different species, and the colored bars are the drugs. And on the y-axis is the difference in resistance between, to that drug-bug combination between LMICs and high-income countries. So I've grouped all the LMICs and all the high-income countries and looked at the difference in resistance. And so anything above 0 tells you that there's a high level of resistance in LMICs. And anything below the bar says the resistance in high-income countries is worse. And so you can see there's only 2 there, and they're both colistin because we only got 1 or 2 isolates. Everything else is worse in LMICs. Just because there's a big bar doesn't mean that the problem's not there in high-income countries. And so I've got an example over on the right there, Acinetobacter baumannii and meropenem. I'll use that as the example. So while there's a huge difference, like 26 percentage points difference between LMIC and high-income country isolates, that's because it's 81.3% level of resistance to meropenem in LMIC isolates. Now, these isolates, remember, were chosen completely blind and usually only 1 per hospital. So this is as random as you can get. The problem's still bad. It's over 50% resistance in high-income countries, but it's just atrocious in LMICs. And so what this shows you is the problem of resistance is really big, especially in LMICs. But you also see, and I'll use Enterobacter cloacae in the middle there with the gray bar, that's ceftazidime-avibactam. That's close to my heart. That was the one I worked on that we got approved when I was at AstraZeneca. You're already seeing erosion in Enterobacter, 11% higher in LMICs. And that's your NDMs, that's your metallo-beta-lactamases that are now getting so hard into Enterobacter cloacae that a lot of them are untreatable. And so that's starting to happen. It's not that, people thought your Cas-Avia resistance would come from KPCs and some of the mutations we saw there down in the loop down at the end. But that's not where it's coming from. It's coming from the metallos that are spreading so widely now. And so LMICs, we know that this was unbiased data that I think put it into sharp relief for at least our funders. So that was good. And hopefully we are about to share this data more widely. The other part of the equation is, of all these resistant isolates, where do all the compounds that CARB-X is funding stand? And so again, it's better to be lucky than good. A lot of the compounds that we have, because they're novel, because they work through different targets and they have different mechanisms of action, we actually are active against them. And so in the left-hand graph is just the Pseudomonas. For people that don't look at these graphs all the time, MIC is along the x-axis and a cumulative percentage on the right. So the steeper the curve and the further to the left it is, the better. And so you can see that for Pseudomonas aeruginosa, the 5 blinded CARB-X funded companies have very good activity against the carbapenem-resistant Pseudomonas and Pseudomonas in general. Same on the right-hand graph here, I've just taken just the low and middle income country Enterobacteriaceae to make it a little bit more sharper because that's where a lot of the carbapenem resistance is. Same story, that these novel mechanism drugs are actually active against the carbapenem resistant Enterobacteriaceae. So that's good, but in vitro activity is the first step of a very long journey to getting a drug to man. I've talked about the safety risks that we did with the University of Queensland. They profiled. Safety departments don't like publishing their data. Sadly, even when I was at AstraZeneca, the tox people in the safety assessment group, it was seen as a competitive advantage. If we found a toxicity signal with a particular class of drug, they were not allowed to publish it because that might be giving a competitor some information. We want them to fail and take 3 years to fail because otherwise they'll catch up faster. And that's a really sad state of affairs where you want other people to fail, because I can tell you when your child or your mum's sick, you don't care who's made the drug that saves their life. And I think it takes a village to raise a child, and we are at a state now within the global society where it doesn't matter who makes the drug, we just need some. So we work with Queensland to take control antibiotics plus theirs through a whole range of different nephrotoxicity models, preclinical models, to try and work out what's the best one that translates to nephrotoxicity in humans, because we know the clinical outcome of some of these drugs. The last one I'll touch on is the antigenic variability. I mentioned that a lot of the companies don't have that expertise or resources, and so we stepped in. I had a colleague that actually was at AstraZeneca with me, is now at CARB-X, runs the vaccine and preventative portfolio at CARB-X. And so he came to me with this problem. He knew where my interests were in bioinformatics and said, is there anything you can do? And I had quite a collection of genome sequences available, and we've been working to supplement those with isolates from areas where there's not a lot of information. And, by that, you can think sub-Saharan Africa, because there's not a lot of surveillance and information coming out of there. And so we have assembled a large number of curated genomes into a database, as geographically diverse as we can. And then we are now partnering with multiple groups to expand that and get more contemporary isolates from those countries of need, and understand what epidemiological clones are circulating there, so that what our vaccines need to hit. What I didn't see early, I thought if you give the bioinformatics analysis to these companies, that would be enough to, they can then map their epitopes and see how conserved they are. Without exception, they came back and said, can you get me that strain? Because I want to test that strain in my animal model. And I hadn't thought that would happen I don't know why, I was naive, I guess. But now what we're doing is we're trying to work to secure those isolates that have those novel antigenic variants and make them available where possible. And that's where the overlaps come. So if groups that we provide sequences, strains to in the therapeutics portfolio, if they sequence those isolates, they give us the sequences, and we blind them, put them in this database, and then we know which isolates match which sequences. And so they share that back. We CARB-X pays for the isolates, pays for the sequencing. And so then they're happy to give the sequences back. We blind it so that company A doesn't know it came from company B. But, anyone that knows, you get a whole genome sequence. And if you don't know what you're looking for, it's still valuable information. It doesn't give anything proprietary away. We've also used the information to build diagnostic sets of isolates for our diagnostic platforms to test their platform technologies against blinded sets of bacteria. So now that we have the susceptibility information, we will build blinded panels, depending on the indication, and send them to all of our diagnostic companies and say, tell us what this is and what it's susceptible to, and pressure test your platform. And some of them were hesitant to do it. And we said, it's a milestone in your contract now that you have to take this set. We know these organisms. We want to see how your diagnostic platform can detect and identify them and tell us, if they're susceptible. So we're sort of bridging now. We do the customized bioinformatics, but we also provide strains and linkage between the strains and the genomes to the groups to help them work. This is just an example of some of the antigens that we looked at, and you're looking over thousands of isolates, and in yellow you get in the little table at the bottom across I think 5,000 isolates there were only 3 allelic types, and the most common was in 99.7% of the strains. So that's a really good antigen. You go to the other end of the scale, antigen number 6. Across the same number of isolates, there was 109 different variants, and the most dominant variant was only in 23% of the isolates. That's a little bit harder to incorporate into a vaccine that will have global use. And so this is the type of analysis we do, and we share with the team. We don't direct what they do, but we provide the information and let them make the decisions. So what's next for CARB-X? We've built a plane. We're now trying to efficiently fly it we built it while it was flying, and now hopefully it's in the air, and we're trying to keep it up there. We are spending a lot of time positioning the existing portfolio for success with advanced developers, because success shouldn't be measured by, did you get something into phase 1? Success is only if it gets through phase 2, phase 3, and then approved to the patient. And so we're working with advanced developers saying, what do you need that would be attractive for you to fund phase 2 and phase 3? And we'll make sure that our companies can provide that in phase 1. And then we have some laser focused rounds that are ongoing now to try and fill some of these gaps. And I think I've got one slide on that. We've had 2 intakes. The third intake starts in the middle of April, and there's 3 different funding themes, and they're up on the website. And I think there is a slide. No, there's not a slide on them. The funding themes are oral therapeutics, because currently, if you get an ESBL UTI, like a UTI caused by an ESBL bacteria, there is no oral drug you can take. You have to go into the hospital and get an IV antibiotic. For an infection that could eminently be treated as an outpatient, you're taking up a bed. And I think especially acute in Australia, or the hospital, the emergency room ramping and the ambulance ramping that's going on here, the COVID burden that has been put on the health systems around the world, you don't want to, 1, give up a bed to someone that could be taking an oral pill and getting fixed at home, but 2, the biggest correlate to getting a more severe infection is the length of time that you stay in a hospital. And so you want to get out as quickly as possible. And so I think there's a need for oral drugs. Tebipenem didn't get up, which is an oral carbapenem. We can argue the stewardship aspects of an oral carbapenem at a different time, but that didn't get up, and that would have been a drug that you could have used in this space. And so I think there is a need for some workhorse oral drugs that can be used. And so different TPPs, different product profiles, but with the focus on oral. Neonatal sepsis, I mentioned that earlier. So vaccines for neonatal sepsis is the third one, and then is the second one. And then the third one is gonorrhea. And the gonorrhea call is actually cross-pillar. It's therapeutics, preventatives, and diagnostics, whatever you've got against gonorrhea. If it's against gonorrhea, send it in. And that's actually been one of the biggest calls that we've had. So third one, if you've got any of those projects, you can talk to me afterwards or, submit an expression of interest by May. So in summary, there is a significant need for push incentives. There's a lot of groups that are working in this space that can't get funding and then die on the vine because they haven't got funding. And so I think the demand for additional funding is acute. And the critical phase, which is this translational phase between basic research and advanced development, is significantly underfunded. It's small groups that are carrying the burden and they're extremely fragile. I think CARB-X is proud of the stewardship and access restrictions or obligations we put into our contracts, and I hope other companies take that on board. I think diagnostics are important, and they're going to become more important as the trend towards narrow spectrum drugs only increases. And I think that the model of wraparound support, we always say we're more than the money at CARB-X. We give that support, both business development support as well as scientific and technical advice across all the different disciplines. And I think that's very well received by a lot of our funded groups. So I always finish on an acknowledgement slide. This is CARB-X. These are the 92 companies that we have funded. We learn something from all of them, whether they succeed or fail. We learn from them. And so they've all helped build CARB-X to what it is today. And we hope to add more in the future. And these are the people that do the work, both at the management level, the finance level, and then the scientific level. So they're my colleagues. So thank you.